Okay, hi everybody. I am going to go over chapter 9.1 and 9.2, and then I will record a second video for 9.3 and 9.4. So, this is just to help you review plant biology for your upcoming quiz this week. So, 9.1 and 9.2 are about the xylem and the phloem, so let's get started. First thing you need to know is about transpiration, and transpiration is a part of gas exchange in the leaf. So, if you look over at this picture, this can kind of explain what you need to know. In the leaf, um, you've got to have CO2 come in because we're going to use that in photosynthesis to make sugars. And then, along with the process of photosynthesis, you also are going to produce the byproduct of oxygen, which will be exchanged for the CO2. So we need CO2 to come in so we can make sugars, and then when we're making the sugars in photosynthesis, we also make oxygen, and that's going to find its way out. And this gas exchange process is going to occur through these little tiny pores on the underside of the leaf called stoma. And the plural of that is stomata. And the little pink guys that are kind of on the sides of that pore, those are called guard cells. And basically, the guard cells are going to puff up when there's a lot of water and they close. I'm sorry, they're going to puff up when there's a lot of water and they open. And then when there's not a lot of water, they become quote unquote flaccid and then they close. So basically, guard cells are going to regulate how much gas exchange can occur based on how much water the plant has. When there's plenty of water, the stoma swell and the, the the guard cells swell and the stoma opens and CO2 is allowed in and oxygen is allowed out. And when there's not a lot of water, then the guard cells become flaccid and the stoma close. And less CO2 is allowed in and less oxygen is allowed out. And the reason for this is that every single time these stoma open, water is also lost from the plant to the atmosphere, and that water loss is called transpiration. So, you all are familiar with the term evaporation. Evaporation is just water that's liquid going into gas form, like maybe evaporating off of a puddle or something like that. But transpiration specifically is evaporation from plants. So plants have water inside of them, and when the stoma open, the liquid water inside of them becomes gas and it's lost to the environment. And plants want to decrease how much transpiration they have occurring because plants have a difficult time coming by water in comparison to animals. So, because water is such a precious commodity to them, they don't want to transpire too much. Because, like we said, we need CO2 in order to do photosynthesis, but we also need water to do photosynthesis. Water is the thing that you break into hydrogens and oxygens, and we need the hydrogens to do the hydrogen pumping, which will help to change ADP into ATP, which is then going to fuel the light independent reactions of photosynthesis. So that's important that we don't lose too much water. But it's just a side effect of being able to collect CO2. So that's the gist on transpiration and understanding guard cells and stomata and gas exchange. The other thing this picture is showing right here is talking about capillary action, which you guys I'm sure are familiar with. Capillary action occurs because water is a polar molecule and water is cohesive, which means that water molecules are attracted to water molecules through hydrogen bonding. And water is also adhesive, which means water molecules are attracted to non-water things like the inside of this glass because the inside of the glass is partially charged. And together, cohesion and adhesion cause the property of capillary action, which allows water to climb up these thin tubes because it's attracted to itself. So it forms the water column, and it's also attracted to the inside of that glass tube, so it climbs up that water column. And the xylem is basically just a really, really thin water, thin tube. which allows water to climb up. Okay, so you guys should just know the term potometer. A potometer is basically just a mechanism that allows us to stick a plant in and then be able to see how much water it's using. So there's a drawing of it that I think is a little bit better right here. So you have a piece of a plant, it's got a stem, and it's got this airtight seal, and you fill this tube with water, and the tube has graduations on it, and it's got a little bubble in there, so then you can tell as the bubble's moving that water is being used or pulled up by the plant, and the plant is transpiring. And under different conditions, like really humid conditions, the plant will transpire less because there's already more water in the atmosphere. And then under really dry conditions, the plant will transpire more because there's not a lot of water in the atmosphere, and the atmosphere is going to pull water from the plant to try to compensate for that. So you just want to know the term potometer, because if you just heard the word potometer, you might not know that it has to do with plants. Okay, so what's special about the xylem is that it has a very, very low pressure, and that's important because things like to flow from high pressure to low pressure. So if you're trying to convince water to go from the soil into your plant, then you want to have a very low pressure inside the xylem and a higher pressure outside of the xylem, so water will flow from the soil into the xylem. And the way that plants have evolved to have a very low pressure is basically to have these very thick but also very narrow tubes. And the narrow tubes are going to have a very low pressure, but the thick walls are going to stop them from collapsing. If you have something that is hollow, it is likely to collapse on itself. But if you have thick walls that make up this hollow thing, it can kind of hold itself open. So usually if things don't have a lot of pressure, they're going to collapse. Usually pressure keeps things open. But we don't want to have high pressure inside the xylem because then water won't go up the xylem. So it's got to have low pressure but also be strong enough that it doesn't collapse on itself. So the way that plants have overcome this kind of challenge is that the walls of the xylem have this this polysaccharide called lignin. So it's kind of like cellulose. It's the same type of molecule that makes up our paper. And xylem walls and vessels are basically made out of lignin. And so the xylem is going to be formed by these cells that basically grow into place to form the xylem, and then when the plant becomes mature, the xylem cells die, but they leave behind their lignin skeleton, and that helps to strengthen the structure of the xylem. But like we said, the important thing is that the pressure inside the xylem is lower than the atmosphere around it, so water will be, quote unquote, convinced to go up the xylem. Okay, and we already talked about cohesion and adhesion on all of that, so we can skip this part here. Besides the capillary action and the cohesion and adhesion that get the water into the xylem, we also need to constantly be pulling the water up the xylem. So even though transpiration is a challenge that plants have to overcome, it also kind of helps them to keep the water moving in the xylem. So if you imagine that you've just got a chain of water molecules all kind of holding hands in a water column, the very top guy in that chain that's closest to the top of the plant, when he evaporates, when he or she evaporates because of transpiration, he gets pulled into the atmosphere to become a gas. But because he is cohesive to the other water molecules, they will all move up a little bit too. And that helps to pull water up that column. So water itself is already attracted to other water molecules, and it's attracted to the inside of the xylem. And then when you have this transpirational pull, where the top water molecules are getting pulled into the air by evaporation or transpiration, it pulls the rest of the column as well and keeps the water moving up the xylem. So it just says here the low pressure generates a pulling force that's transmitted through the water in the xylem vessels down the stem and to the ends of the roots. And it's strong enough to get the water to continuously move. Not at a super fast rate, but it does continuously move. And water is unique because it's able to do this without breaking that water column because of its strong cohesive properties. So a reminder, you also need to know lots of properties of water from chapter 2.2. The other thing that they talk about in this chapter is about minerals in the roots, because it's not just water that we need to transport into the plant. We also need to get minerals from the soil. And the way that that's going to happen is mostly through active transport. So there's protein pumps in the root cells of plants that pump different ions and minerals from the soil into the plant. So maybe things like phosphorus or magnesium, those are going to have protein pumps that transfer them from the outside soil into the root cells. And then if you're pumping things like magnesium or phosphorus or nitrogen from the soil, water is going to follow by osmosis. So we have active transport of the ions, but we have passive transport of the water. This way, we're not using a ton of energy, though we are using energy, and the plants are able to have glucose and ATP to provide that energy. This little section right here is just talking about some plants. They have fungi which grow on the surface of the roots, and the fungi are going to help to absorb mineral ions, because sometimes the plants, because they have to actively transport the ions, and plants are not particularly a high energy class of species, sometimes that ion transport on their own, doing active transport and pumping the ions from the soil on their own, is not as efficient as having another organism help you out. So that's just an example of a mutualistic relationship, because the fungi that grow on the roots of these plants are transporting more ions for them into the plant that are needed for growth, and the plant will feed the fungi from the glucose or sucrose that it makes through photosynthesis. Okay, just scrolling along. And then we already talked about transpiration, so you all should understand what transpiration is. And this last section here is just talking about these two pathways that exist in a plant. They're called the apoplast pathway and the symplast pathway. If water is going from cell wall to cell wall, then it's called the apoplast pathway. And if it's going through the cytoplasm of one plant cell to the next plant cell, that's called the symplast pathway. And there is a picture right here. That's something that you might want to be familiar with for the IB exam in a couple of weeks, but it is not something that you need to know for this quiz that Ms. Cunningham wrote for you all for this week. And then this next section is talking about adaptations for water conservation. Like we said, it's really important for plants to hold on to water as best as they can, because they are not able to move to sources of water and drink water like animals can. So they have to deal with the water that they have, and rainy seasons or dry seasons, they have to just try to persevere. So some plants have evolved adaptations for water conservation. One class of plants are called xerophytes, and they're plants that grow in deserts and other dry habitats. So one of the things that they do to try to increase their water retention is that they will have a dormant period, and their embryos will not grow until it rains, to say that there's going to be enough water to support their growth from an embryo into an actual plant. And then cacti are an example of xerophytes. And basically cacti have leaves, but they're so small and reduced that we really know them better as spines. And the spines are smaller, or the leaves are smaller, which is going to decrease the number of stomata they have. So it's also going to decrease the amount of water loss that they can have. Because like you remember, the stomata are the holes or the pores on the underside of the leaf that let in CO2, let out oxygen, but also let out water. So if you reduce your leaves into spines, then you have reduced the surface area to volume ratio of possibility for transpiration. So that's one of the adaptations. A couple of years ago, maybe in 2017, maybe 2018, I can't quite remember, one of the long questions for writing at the end of paper 2 was asking about the different adaptations that plants have for conserving water. So this box here would be really important for that. So like we said, some plants will have, instead of leaves, what we call reduced leaves or spines. Other plants will do this thing where the stomata only open at night because at night it'll be less warm, so you will lose less water through transpiration. Other plants will do this thing where they collect carbon dioxide and they convert the carbon dioxide into another molecule called malic acid. So when the day comes, they can reconvert that malic acid back into the carbon dioxide they need for the process of photosynthesis, and they won't lose it through gas exchange. So this is kind of a good page to read, this 410 in your textbook. And then there's a little list here about plants that live in really salty environments called halophytes and what kind of adaptations they have to try to save water in the environment that they exist in. So of chapter 9.1, I would say page 410 is probably the most important for the purposes of the IB exam, but I do not think that this information is super important for this week's quiz. You guys are also supposed to be technically able to draw and label xylem vessels or identify them from a cross-section. So here's a classic cross-section of a plant, and what you guys should notice is that you've got your xylem here closer to the center of the stem, and then you've got your phloem closer to the perimeter or circumference of the stem. And then there is a set of tissue called cambium that separates the xylem and the phloem, and together the xylem, cambia, and phloem are called the vascular bundle. But again, the xylem is closer to the center of the cross section and the phloem is closer to the outside of the cross section. So there are just some pictures here, right here. This is the xylem, here's the cambium, and here's the phloem closer to the outside. This picture here is just showing the lignin, which allows the xylem to be strong and for water to go up. And that's basically 9.1. Then we want to move on into 9.2, which is about the phloem. I think 9.1 is easier to understand, and the stuff in the phloem is a little bit more complex. So when we're talking about the phloem is for the transport of nutrients. Particularly, we're going to talk about sucrose, which is a disaccharide sugar. And the xylem, as you know, transports water. So when you're transporting sugars, we call it translocation. And the translocation of sugars in the plant occurs from what we call sources to sinks. Sources are places where the sugar is made, and sinks are places either where the sugar is going to be stored or places where it's going to be used up. And the phloem is not made out of the lignin, it's made out of these cells called sieve tube cells. And the sieve tube cells are going to be connected to each other by these walls that have holes in them called sieve plates. And we've also got these other cells that are next to the sieve tube cells, which are called companion cells. And they help each other out, so it should make sense that they are the companion of the sieve tube cells. So unlike the xylem, which is unidirectional, always moving from root to stem to leaf to atmosphere, the phloem is going to be bidirectional, so things can move up or they can move down the phloem, depending on which direction the source is and where the sink is. So the phloem is going to try to transport things from the place where you do have sugars and amino acids and other important molecules to places where you don't have sugars or amino acids and you need those in order to grow and survive. There's a table on the next page that shows which places are sinks and which places are sources, and we'll talk about that next. It's not really a fixed thing, but these are the general rules. So sources would be places where you're making sucrose, would be where you do photosynthesis, obviously, in your leaves, and then in your stems, photosynthesis occurs. Those are sources. Places where you've stored sugars and now you can use them would also be sources. So when you talk about a seed that is about to germinate, it's got a big chunk of sugars in there, and that's a source because the growing plant is going to pull the nutrients from the rest of that seed to try to grow. If you have tap roots or tubers, so like we're talking about like a potato, those are going to be sources as well because as the rest of the plant grows, they're going to try to take sugars away from those tap roots or tubers. And then we've got sinks. And sinks are going to be places that are going to absorb the ions, and they're going to absorb ions, or they're going to absorb the sugars, and they need these things in order to grow. So if you're developing fruit, you're going to pull sugars. If you're developing seed, you're going to pull sugars. If you're growing leaves, you're going to pull sugars. If you're developing taproot, you're going to pull sugars. And then we can see that the developing taproot, once it's developed, it will become a source. And growing leaves, once they're fully grown, they'll become mature leaves and they'll become a source. So this is a pretty easy concept to get. Okay, so the next thing is phloem loading. Phloem loading, is the general term for the active transport that we use or plants use to take organic compounds such as sucrose into the phloem sieve tubes. And it can happen in a couple of different ways. Like before, we were talking about the apoplast route, and we were talking about the symplast route. For the apoplast route, it's going to be based off of a hydrogen or proton gradient. So hydrogens, or H pluses, hydrogen ions, are also known as protons, and we have seen a ton of times, lots of examples in IB Bio where proton gradients are used to transport stuff or make stuff happen. You have a proton gradient in the electron transport chain of cell respiration. You also have a proton gradient in the light-dependent reactions of photosynthesis. Okay, so what we're seeing here, we basically, in the cell wall of the sieve tube cells, you have a proton pump that is going to pump hydrogen ions or protons from one side of the cell to the other side of the cell. And it's going to move from low concentration to high concentration. So that's why we have to use a pump, because it is active transport. So we move hydrogens against their concentration gradient into an area they don't really want to be in. Then we have another protein that's embedded in the cell wall, and this is going to be a co-transporter. And basically what it does is it moves sucrose from one side to the other side along its concentration gradient and also along the hydrogen's concentration gradient. So by pairing these two low energy pathways together, then we are able to transport the sucrose without using too much energy. So this is another good picture to look at, but really I don't think that you guys need to know it, particularly for Ms. Cunningham's test, and this is kind of a too specific thing for them to ask a lot about on the IB exam. So moving on, we're looking at the symplast and apoplast routes. So apoplast is going through the cell walls, and then symplast is going through the cytoplasm of cell to cell to cell. And each of the cells has these things called plasmodesmata, which are basically like gaps in the cell wall between the plants, and it allows the plants to communicate and share materials with each other. And in the symplast route, basically we still keep a concentration gradient for where we have high sucrose over here and low sucrose over here. And the way that we do that is once the sucrose makes its way to the end of the chain and it's finally in the sink, then it's going to be converted into an oligosaccharide. So we keep that high concentration gradient. So we can't just move sucrose here to here to here to here to here and then not do anything with it at the end. Because if we do that, then eventually there won't be a gradient anymore. It'll even out. So if we keep changing the sucrose at the end into something else, then we'll keep the concentration of sucrose over here low. This again is not really a concept that I think you guys need to know that well, but you should know the concept of the fact that we like concentration gradients. They help us to transport things from high to low without using too much energy. So we want to keep that kind of concentration gradient going. Okay. So here it's just talking about pressure and water potential differences playing a role in translocation, and we know that pressure gradients and water potential differences are good things. It's kind of just like we were talking about. We always like to move things from high to low. It's just the natural movement of things in biology and chemistry on Earth. So we'd like to keep those gradients going. So when you have sucrose and other carbohydrates being put into the companion cells, it's going to draw water with them through osmosis because water likes to follow solutes. And then because water is following where these solutes are going, it ends up building up pressure. And the water in that cell is then going to flow from areas of high pressure to areas of low pressure. And like we said, on the end where we have the sink, the sucrose is going to be pulled from the phloem and it's going to be used as an energy source, either like letting that area grow or going to convert that sucrose into starch where we can store it. In either case, what we're doing is we're changing the sucrose into something else, and that's going to reduce how much pressure there is. And then if we get to reduce how much pressure there is, it keeps things moving because we're still maintaining the fact that we're going from a higher to lower pressure instead of the pressure building up so that it's just high to high pressure. And if that happens, nothing's really moving. And then we are talking about phloem sieve tubes, which are the cells that mostly make up the phloem. And unlike in the xylem, they are living and they've got the companion cell, which helps them out. And in the xylem, we have a lot of water moving through passive transport. However, in the phloem, we have a lot of things moving through active transport. And what we can see in this next picture is that the companion cells that go with the sieve tube cells, they have a lot of mitochondria because of all the active transport that needs to be done. This orange box here is a lot of a repeat of what we've already talked about how translocation occurs, so you don't really need to take a look at this box too much. So here's a companion cell, and all these dark circles, those are all mitochondria, which are helping with the active transport of sucrose. This purple box here, it was asked a while ago, I think in 2016 on a test. So they're not going to ask about it again because it's too soon, but it's just saying that in the past they've been able to use the stylets of aphids, which is basically the little part of the aphid that it uses to try to suck the phloem sap out of plants. You can use that to try to draw out some of the phloem, and you can, in the lab, analyze the contents of the phloem. So you can take a look at this later, but it's not going to be on your quiz this week. And I think that almost brings us to the end of 9.2. This other stuff is just DBQ stuff. And again, you guys should be able to identify the difference between xylem and phloem. The xylem is usually going to be bigger. So here's the xylem here, and the phloem's over here. And the reason why the xylem is bigger is because the xylem is made out of water, and water is cohesive, or not, the xylem's not made out of water, the xylem carries water, and water is cohesive. And because water has cohesive properties, it doesn't need such narrow tubes to stick together because it already has a affinity to sticking together. And then over here we have another cross section. Again, closer to the inside is the xylem. So all of these like white circles that you see are the xylem. And then this ring of kind of pinkish purple on the outside, those are going to all be the phloem. And the blue in between them, that's the cambium. And together they make a vascular bundle. So you should just kind of be familiar with that. And then there's just like a bunch of different pictures of different cross sections. This one does not look as neat, but still, just take a look at those for future reference, but again, will not be on your quiz for this week with Ms. Cunningham. Okay, so that's 9-1 and 9-2, and I'm going to pause there